What if the plants growing in your backyard, in the forest behind your house, and in the spice rack on your kitchen counter were doing something that the most advanced antibiotics cannot? Not because they are more powerful, but because bacteria simply cannot figure out how to beat them. Pharmaceutical antibiotics work through a single compound with a single target. Bacteria are extraordinarily good at evolving around single targets. And they have. But plants produce dozens of active compounds simultaneously, attacking bacterial cell walls, disrupting biofilm, blocking bacterial communication, and activating immune response all at once. There is no single mutation that makes a bacterium resistant to all of that at once. Today I am walking you through 20 of those plants, starting with one that is virtually unknown in the Western world despite documented activity against MRSA and drug-resistant malaria. And number 4 is an oil whose carvacrol content surprised the researchers when they tested it against clinical MRSA isolates. Stick around because number 9 belongs to the same plant genus that won the Nobel Prize for antimicrobial discovery. And it has been fighting infections in the human gut for thousands of years before anyone knew why. Number 1. Cryptolepis sanguinolenta, the West African antimicrobial plant, virtually unknown in Western herbal medicine, despite documented activity against Plasmodium falciparum, the malaria parasite resistant to pharmaceutical chloroquine, and against MRSA through a mechanism involving cryptolepine's DNA intercalation that creates a fundamentally different challenge for bacterial resistance development. Cryptolepis sanguinolenta is a woody vine native to West Africa that has been used in traditional medicine across Ghana, Nigeria, and Sierra Leone for generations to treat malaria, typhoid fever, and bacterial infections. The alkaloid cryptolepine, the primary bioactive compound, has attracted serious pharmaceutical research attention for 2 reasons that are rarely found together in a single plant compound. First, it has documented activity against chloroquine-resistant Plasmodium falciparum, the malaria strain that pharmaceutical resistance has made the most difficult to treat. Second, it has documented antibacterial activity against MRSA through DNA intercalation, a mechanism where the compound inserts itself into the DNA helix of bacterial cells, disrupting replication. This mechanism is fundamentally different from how conventional antibiotics work, which is precisely why bacteria that have evolved resistance to standard antibiotics are still vulnerable to cryptolepine. Clinical research in Ghana has studied Cryptolepis preparations for uncomplicated malaria with outcomes comparable to chloroquine in drug-sensitive cases. A plant used for generations by traditional healers in West Africa is now being studied by pharmaceutical researchers as a model for next-generation antimicrobials. It remains almost entirely unknown in Western herbal medicine, not because the evidence is absent, but because the commercial infrastructure to make it available has not yet caught up to the research. Number 2 grows on trees and rocks in forests, hanging in gray-green strands from branches, and has been called the wilderness antibiotic, the plant that traditional herbalists and survival medicine practitioners reach for when nothing else is available. The compound it contains was studied as a potential pharmaceutical antibiotic before the research was abandoned for reasons that had more to do with economics than efficacy. Number 2. Usnea, the lichen that grows on trees across temperate forests worldwide, containing usnic acid with documented broad-spectrum antibacterial activity, studied as a pharmaceutical antibiotic candidate before being abandoned due to formulation challenges rather than lack of efficacy, and used by traditional healers across multiple continents as a wound antimicrobial and respiratory infection treatment. Usnea is not technically a plant. It is a lichen, a symbiotic organism combining fungal and algal components. But it has been used in herbal medicine across North America, Europe, and Asia for centuries as one of the most reliable antimicrobial preparations available from wild sources. The primary active compound usnic acid has documented antibacterial activity against gram-positive organisms, including Staphylococcus aureus, Streptococcus species, and Mycobacterium tuberculosis in laboratory research. It was studied as a pharmaceutical antibiotic candidate in the mid-20th century, and the research confirmed genuine antimicrobial potency. The development was abandoned not because usnic acid did not work, but because its poor water solubility created formulation challenges that made it difficult to produce as a standard oral pharmaceutical. The antimicrobial activity remained. the pharmacological interest remained. What did not remain was the pharmaceutical investment. Traditional herbalists never stopped using it. Usnea tincture, prepared by extracting the lichen in high-proof alcohol, which effectively addresses the solubility issue, has been used for upper respiratory infections, urinary tract infections, and wound care. In wilderness medicine and survival contexts, fresh Usnea packed into a wound delivers usnic acid directly to the site of infection. The forest pharmacy that pharmaceutical investment walked away from is still growing on every oak tree in the temperate world. Number 3. Andrographis paniculata, the Indian echinacea, whose primary compound andrographolide has been studied in randomized controlled trials for upper respiratory infections, with results showing significant reduction in symptom severity and duration comparable to antibiotic treatment for uncomplicated sinusitis and bronchitis, with a mechanism that includes direct antimicrobial activity alongside immune modulation. Andrographis paniculata is one of the most widely used medicinal plants in Ayurvedic and traditional Chinese medicine, prescribed specifically for acute infections, fever, and inflammatory conditions for thousands of years. The primary active compound andrographolide has attracted more clinical research than most Western herbal practitioners realize. A systematic review of randomized controlled trials published in the Journal of Clinical Pharmacy and Therapeutics found that andrographis supplementation significantly reduced upper respiratory infection symptom severity and duration compared to placebo. More significantly for the antibiotic resistance context, a Swedish randomized controlled trial found that an andrographis preparation was comparable to antibiotic treatment for uncomplicated sinusitis, a condition for which antibiotics are routinely overprescribed and for which antibiotic resistance is actively developing. Andrographolide achieves its effects through multiple mechanisms: direct antimicrobial activity against several bacterial species, inhibition of NF-κB inflammatory signaling that reduces the inflammatory damage associated with infection, and enhancement of immune cell function including natural killer cell and macrophage activity. The bitter taste of andrographis, extreme bitterness, is the first thing anyone notices, is itself part of the mechanism, stimulating digestive immune function through bitter receptor activation in the gut lining. Number 4. Oil of oregano, whose primary compound carvacrol demonstrated activity against clinical MRSA isolates in laboratory studies that surprised researchers expecting a modest effect, operating through a mechanism of bacterial cell membrane disruption that simultaneously addresses multiple cellular functions and creates a resistance challenge that single-target antibiotics do not pose. Origanum vulgare and related species produce carvacrol and thymol, volatile phenolic compounds that give oregano its characteristic aroma and that have attracted serious antimicrobial research. The MRSA finding is the one that most surprises people. Carvacrol has demonstrated activity against clinical MRSA isolates, the bacteria collected from actual infected patients, not laboratory strains, with minimum inhibitory concentrations that compare favorably to some pharmaceutical antibiotics in vitro conditions. The mechanism explains why bacteria have difficulty developing resistance. Carvacrol attacks the bacterial cell membrane, increasing its permeability and disrupting the ion gradients that membrane integrity maintains. Simultaneously, it interferes with bacterial biofilm formation, the protective matrix that bacteria construct to shield colonies from antimicrobial attack. And it disrupts bacterial quorum sensing, the chemical communication system that bacteria use to coordinate collective behavior. Addressing membrane integrity, biofilm formation, and quorum sensing simultaneously through a single compound is the kind of multi-target mechanism that makes resistance evolution significantly more difficult. High-quality oregano oil should be standardized to carvacrol content, the active fraction, and must be significantly diluted before any use, as concentrated carvacrol is intensely irritating to mucous membranes. The culinary herb and the therapeutic preparation are related but functionally different products. Number 5. Propolis, the resinous substance that honeybees collect from tree buds and use to seal and sterilize their hives, containing over 300 identified bioactive compounds including flavonoids, phenolic acids, and terpenes with documented broad-spectrum antibacterial, antiviral, and antifungal activity that reflects the evolutionary pressure of maintaining a sterile colony environment. Propolis is not a plant. It is a plant-derived substance collected and processed by bees from the resinous buds of trees, primarily poplar, birch, and pine. Bees use it to seal gaps in the hive, coat the interior surfaces, and mummify intruding organisms that are too large to remove. The antimicrobial requirements of maintaining a honeybee colony, tens of thousands of organisms living in close proximity with food stores that represent a major target for pathogens, have driven the selection of a substance with extraordinarily broad antimicrobial coverage. Over 300 bioactive compounds have been identified in propolis, varying by geographic region based on the resin sources available to local bee populations. The primary antimicrobial constituents include caffeic acid, phenethyl ester, pinosembrin, galangin, and various flavonoids, each with individual antimicrobial activity that together produce a synergistic coverage difficult for any single pathogen to evade. Clinical research has documented propolis activity against Streptococcus mutans, Staphylococcus aureus, including MRSA, Candida species, and multiple respiratory viruses. Propolis tincture has been studied for upper respiratory infections, oral health, and wound care with consistent positive outcomes. The 300-compound profile that evolved to keep a bee colony sterile is available as a tincture, spray, or lozenge at any health food store. A ready-made multi-compound antimicrobial that 300 million years of evolutionary pressure optimized. Number 6. Pau d'arco, Tabebuia impetigonosa, the inner bark of a South American tree used by indigenous Amazonian peoples for generations against fungal infections, containing lapachol and beta-lapachone, with documented antifungal activity against Candida species, resistant to pharmaceutical antifungals and antibacterial activity through quinone-mediated oxidative stress mechanisms. Pau d'arco is the inner bark of several Tabebuia tree species, native to the Amazon basin and Central America. Indigenous use of the bark as a tea for fungal infections, fever, and inflammatory conditions predates contact with Western medicine by centuries. The primary active compounds, lapachol and beta-lapachone, are naphthoquinones that generate reactive oxygen species in target cells, producing oxidative stress that disrupts cellular function. This quinone-mediated mechanism is distinct from the mechanisms of pharmaceutical antifungals like fluconazole, which means that Candida strains resistant to pharmaceutical treatment retain vulnerability to lapachol through different pathways. Laboratory research has documented Pau d'arco activity against multiple Candida species, including fluconazole-resistant strains, against several bacterial species, and against certain parasites. The tea, made from the inner bark, not the outer bark, is the traditional preparation, consumed over extended periods for chronic fungal conditions, including intestinal candidiasis and recurrent vaginal yeast infections. The Amazon has been called the world's largest pharmacy, and Pau d'arco represents the kind of botanical resource that indigenous medical systems identified through generations of observation that Western pharmaceutical research is still working to fully characterize. Number 7. Golden seal, Hydrastis canadensis, containing berberine at concentrations that disrupt bacterial biofilm formation in antibiotic-resistant organisms, modify gut microbiome composition in ways comparable to pharmaceutical interventions, and demonstrate activity against H. pylori in clinical research through a mechanism that operates differently enough from conventional antibiotics that cross-resistance is limited. Golden seal is one of the most important antimicrobial herbs in North American indigenous medicine and remains one of the best-selling herbal supplements in the United States. The primary active compound berberine has attracted more clinical research than most plant compounds, and the findings are consistently more significant than mainstream medicine acknowledges. Berberine disrupts bacterial biofilm formation, the protective matrix that drug-resistant bacteria construct to shield themselves from antibiotic penetration, in strains including MRSA and Pseudomonas aeruginosa. It demonstrates activity against H. pylori, the bacterium responsible for most gastric ulcers, through mechanisms that include inhibition of H. pylori adhesion to gastric epithelial cells. Clinical research has found berberine's effects on gut microbiome composition comparable to pharmaceutical interventions in some metabolic applications, suggesting its antimicrobial activity in the gut is selective enough to reshape the microbial community rather than simply deplete it. The yellow color of golden seal, from berberine itself, stains anything it contacts and has been used as a natural dye. That same color is the signal of the compound's therapeutic potency. Overharvesting has made genuine wild golden seal increasingly scarce. Cultivated sources or berberine-containing alternatives like Oregon grapefruit provide the same active compound from more sustainable sources. If this list is making you think seriously about having these plants available when you need them, not just knowing about them, but actually knowing how to grow, prepare, and use them at home, there is a complete practical system in the first pinned comment below. Step-by-step guides to the most effective medicinal plants for home use, with preparation protocols, dosage notes, and everything you need to move from knowledge to application. Link is pinned below. Now let us keep going. Number 8. Cat's claw, Uncaria tomentosa, the Amazonian vine whose oxandole alkaloids produce immune modulation effects that enhance the body's own antimicrobial capacity while simultaneously demonstrating direct antibacterial activity used by indigenous peoples of the Peruvian Amazon for infections, inflammatory conditions, and immune dysfunction for at least 2,000 years. Cat's claw is a large woody vine native to the Amazon rainforest, named for the curved thorns that allow it to climb forest trees. The inner bark and root contain oxandole alkaloids, particularly pteropodine, isopteropodine, and mitriphyline, alongside pentacyclic alkaloids with documented immunostimulatory activity. Research has confirmed that cat's claw alkaloids enhance phagocytosis, the process by which immune cells engulf and destroy pathogens, and increase the proliferation and activity of natural killer cells and T lymphocytes. This immune enhancement addresses the fundamental limitation of antimicrobial plants used in isolation. They are most effective when the immune system they are supporting is functioning adequately. Cat's claw provides both direct antimicrobial activity against certain bacterial and viral pathogens and the immune modulation that makes the body's own defenses more effective against the pathogens the plant does not directly address. It has been studied for HIV-related immune dysfunction, rheumatoid arthritis, and cancer-related immune suppression, applications that reflect its capacity to modulate immune function in conditions beyond simple infection. Traditional Peruvian use as a tea from the inner bark for chronic infections, inflammatory conditions, and immune weakness is consistent with the multi-mechanism profile that modern research has confirmed. Number 9. Wormwood, Artemisia absinthium, and related species. From the same genus that produced artemisinin, the Nobel Prize-winning anti-malarial compound discovered by Tu Youyou in 2015, containing sesquiterpene lactones with documented antimicrobial activity against intestinal parasites, H. pylori, and Candida through mechanisms that include disruption of the mitochondrial electron transport chain. In 2015, Chinese scientist Tu Youyou received the Nobel Prize in Physiology or Medicine for discovering artemisinin, the anti-malarial compound derived from Artemisia annua. That award validated something traditional medicine systems had known for 2,000 years, that Artemisia species contain compounds with extraordinary antimicrobial and antiparasitic potency. The absinthin and artemisinin sesquiterpene lactones in Artemisia absinthium, common wormwood, have documented activity against intestinal parasites, H. pylori and Candida species, through mechanisms that include disruption of the mitochondrial electron transport chain in target organisms, producing the cellular energy crisis that kills the pathogen. Wormwood has been used continuously across European, Middle Eastern, and Asian traditional medicine for intestinal worm infestations, digestive infections, and febrile illness for thousands of years. The bitter compounds simultaneously stimulate bile production and digestive enzyme secretion, creating the alkaline, enzyme-rich digestive environment that is itself inhospitable to many intestinal pathogens. Short-term therapeutic use of wormwood tea or tincture for documented intestinal parasitic infection, or H. pylori, is supported by both traditional use and research. Long-term use of large amounts is not appropriate due to thujone accumulation. The genus that won the Nobel Prize for Antimicrobial Discovery had been fighting infections in the human gut for millennia before the prize committee caught up. Number 10. Garlic, Allium sativum, containing allicin whose antimicrobial mechanism operates through irreversible inhibition of bacterial thiol enzymes, a mechanism that bacteria cannot easily develop resistance to because the same enzymes are essential for fundamental cellular processes, with documented clinical activity against H. pylori, MRSA, and multiple drug-resistant organisms. Garlic has been used as medicine for at least 5,000 years across virtually every culture that had access to it. The antimicrobial mechanism of allicin, the compound produced when garlic is crushed, allowing alliinase to convert alliin, is fundamentally different from how antibiotics work. Allicin reacts with and permanently disables thiol-containing enzymes in bacterial cells. These enzymes are involved in critical cellular processes that bacteria cannot simply evolve around. The enzymes are too important to the organism's survival to be discarded, and allicin's reaction with them is irreversible. This creates a mechanistic barrier to resistance development that single-target antibiotics lack. Laboratory research has documented allicin activity against MRSA, Pseudomonas aeruginosa, Escherichia coli, Salmonella, and multiple other pathogens, including drug-resistant strains. Clinical research has found garlic preparations active against H. pylori in human subjects. The 10-minute waiting period after crushing garlic before cooking or consuming it maximizes allicin formation, a preparation detail that makes the difference between garlic as seasoning and garlic as antimicrobial. Aged garlic extract provides different compounds with different activity profiles, useful for immune modulation and cardiovascular support, but less active against acute infection than fresh crushed garlic. Number 11. Thyme, Thymus vulgaris, whose primary compound thymol is the active ingredient in Listerine mouthwash and in pharmaceutical antiseptic preparations. With documented antibacterial and antifungal activity confirmed across multiple research centers, and with the telling historical detail that thymol was the primary pharmaceutical antiseptic before synthetic alternatives were developed. Thyme has been used as a medicinal herb since ancient Egyptian and Greek civilizations. It was used in embalming preparations in ancient Egypt and prescribed by Hippocrates for respiratory infections. The primary volatile compound thymol has a history in pharmaceutical medicine that most people do not know. It was the primary active ingredient in Listerine when the mouthwash was first formulated in 1879. It was used as a pharmaceutical antifungal for hookworm and other intestinal parasites before synthetic alternatives were developed, and it remains an active ingredient in several pharmaceutical antiseptic preparations today. The fact that thymol was replaced by synthetic alternatives was not because it stopped working. It was because synthetic compounds could be patented and the natural compound could not. Laboratory research consistently confirms thymol's broad-spectrum antibacterial and antifungal activity against respiratory pathogens, oral bacteria, foodborne pathogens, and Candida species. Thyme tea for respiratory infections. The traditional preparation across European folk medicine delivers therapeutic concentrations of thymol to the respiratory mucosa through both direct contact and volatile compound inhalation during preparation. The herb that became the original antiseptic ingredient is still growing in kitchen gardens everywhere. Number 12. Elderberry, Sambucus nigra, whose primary flavonoids inhibit neuraminidase through the same mechanism as oseltamivir. Tamiflu, the pharmaceutical antiviral that costs over $100 per treatment course, with multiple meta-analyses of randomized controlled trials confirming significant reduction in influenza duration and severity. The elderberry's mechanism of action against influenza was identified when researchers examining its flavonoids, particularly cyanidin-3 glucoside and cyanidin-3 sambubioside, found that they bind to and inhibit neuraminidase, the enzyme that influenza viruses use to release newly formed viral particles from infected cells and spread to uninfected tissue. This is precisely the mechanism of oseltamivir, sold as Tamiflu, the pharmaceutical antiviral prescribed for influenza that costs over $100 per 5-day treatment course. A meta-analysis of randomized controlled trials published in Complementary Medicine Research found that elderberry supplementation significantly reduced the duration and severity of influenza symptoms compared to placebo. The neuraminidase inhibition mechanism means that elderberry's antiviral activity is not nonspecific immune stimulation. It is a targeted molecular interaction with a specific viral enzyme, the same enzyme that pharmaceutical antiviral development has focused on. Victorian households made elderberry syrup every autumn from wild harvested berries and stored it through winter as the primary influenza preparation. Modern research has confirmed they were producing a preparation with documented antiviral activity against the exact pathogen that most threaten them each winter season. Number 13. Olive leaf, Olea europaea, containing oleuropein with documented antiviral activity against influenza A and B, herpes simplex viruses 1 and 2, parainfluenza, and in laboratory research retroviruses, alongside antibacterial activity against multiple gram-positive and gram-negative organisms through a mechanism involving disruption of viral envelope proteins and bacterial membrane integrity. The olive tree is mentioned over 500 times in Scripture and revered across Mediterranean cultures for thousands of years. While olive oil attracts most attention for its therapeutic properties, the leaves of the same tree contain a distinct and therapeutically significant compound profile. Oleuropein, the primary sequiridoid in olive leaves, has attracted significant antiviral research attention for the breadth of its documented activity. Laboratory studies have confirmed activity against influenza A and B, herpes simplex viruses 1 and 2, parainfluenza virus, Epstein-Barr virus, and in research models, retroviruses. The mechanism involves interaction with viral envelope proteins, disrupting the structural components that viruses use to recognize and enter host cells. Antibacterial activity against gram-positive and gram-negative organisms has been confirmed across multiple research settings. Olive leaf has also demonstrated AC-inhibiting activity that contributes to blood pressure reduction, a cardiovascular benefit that is secondary to its antimicrobial profile but significant for the population most affected by chronic infection. Traditional healers in Greece, Italy, and across the Mediterranean world used olive leaves medicinally while using the oil culinarily, correctly identifying that these were two different preparations from the same plant with different therapeutic applications. Number 14. Tea tree, Melaleuca alternifolia, whose primary compound terpenine 4-ol is used in certified clinical wound dressings in hospital settings, with documented activity against MRSA, Candida species resistant to pharmaceutical antifungals, and biofilm-forming organisms, and the specific detail that its mechanism of membrane disruption is difficult for bacteria to develop resistance to. Tea tree oil from Melaleuca alternifolia has been used by Aboriginal Australians for wound healing and skin infections for thousands of years. The primary active compound terpenine 4-ol has the unusual distinction of appearing in products that have obtained regulatory approval for clinical use in hospital settings. Medical-grade tea tree oil wound dressings are available in several countries for the management of MRSA-colonized wounds and surgical site infections. The mechanism involves disruption of bacterial and fungal cell membranes, similar to oregano's carvacrol but through different specific interactions with membrane lipids. This membrane disruption mechanism applies to Candida species, including fluconazole-resistant strains, creating antifungal coverage where pharmaceutical options are failing. Importantly, research has found no documented resistance development to terpenine 4-ol in MRSA strains exposed to therapeutic concentrations, a finding consistent with the mechanism that makes resistance development structurally challenging. Tea tree oil must be used topically and significantly diluted in a carrier oil for skin application. Oral consumption is not appropriate and can cause toxicity. The topical antimicrobial that was known to Indigenous Australians for generations is now validated by clinical research and used in hospital wound management. Number 15. Licorice root, Glycyrrhiza glabra, containing glycyrrhizin and glycyrrhizinic acid, with documented antiviral activity against herpes simplex viruses, hepatitis C, respiratory syncytial virus, and SARS-related coronaviruses in laboratory research, alongside direct antimicrobial activity against H. pylori comparable to triple antibiotic therapy in some clinical comparisons. Licorice root has been used medicinally for over 4,000 years across Ayurvedic, Chinese, and Middle Eastern traditional medicine. It appears in the Ebers Papyrus and in ancient Chinese medical texts as one of the most widely prescribed herbs in those systems. The primary active compound glycyrrhizin and its metabolite glycyrrhizinic acid have attracted antiviral research across multiple pathogen categories. Laboratory studies have documented activity against herpes simplex viruses 1 and 2, hepatitis C virus, respiratory syncytial virus, and, in research conducted following the SARS outbreak, SARS-related coronavirus. The mechanism involves interference with viral replication pathways, including inhibition of viral protein synthesis and disruption of viral envelope formation. For H. pylori specifically, deglycyrrhizated licorice, DGL, the form with the blood pressure-raising glycyrrhizin removed, has been studied in clinical comparisons with standard triple antibiotic therapy and shown H. pylori eradication rates that, in some trials, were comparable to the pharmaceutical standard of care. Licorice root in its whole form cannot be taken long-term due to glycyrrhizin's aldosterone-mimicking effects that raise blood pressure and deplete potassium. The DGL form removes this concern while preserving most of the therapeutic activity. Number 16. Echinacea. Echinacea purpurea and related species, whose alkylamides activate cannabinoid receptors in immune cells to enhance phagocytosis and cytokine production, with meta-analyses of clinical trials finding significant reduction in the incidence and duration of upper respiratory infections and the specific finding that it reduces the likelihood of a cold developing after viral exposure. Echinacea is the most commercially successful medicinal herb in the Western world and simultaneously one of the most misunderstood. The mechanism is not vague immune boosting. The alkylamides in Echinacea root and aerial parts bind specifically to CB2 cannabinoid receptors on immune cells, activating signaling pathways that enhance phagocytosis, increase natural killer cell activity, and modulate cytokine production. This is a specific receptor-mediated mechanism, not a non-specific enhancement. Meta-analyses of clinical trials have found that Echinacea preparations significantly reduce the incidence of colds when taken during exposure periods and reduce duration when taken at onset, a specific finding that distinguishes Echinacea from simple symptomatic treatments. It reduces the probability of a cold developing after documented viral exposure, meaning it is interfering with the infection establishment process rather than merely managing symptoms. The quality variation in commercial Echinacea products is enormous. Aerial parts of the plant have different active compound profiles than the root, and different species have different activity profiles. Echinacea purpurea aerial parts standardized for alkylamide content, or Echinacea angustifolia root, represent the preparations with the most consistent clinical evidence. Number 17. Clove, Syzygium aromaticum, whose primary compound eugenol is still used in modern dentistry as a local anesthetic and antiseptic in clinical formulations, with laboratory studies confirming broad-spectrum antibacterial activity against over 20 bacterial species and antifungal activity against Candida through membrane disruption mechanisms. Clove has been used as both a culinary spice and medicinal plant across Asian, Middle Eastern, and eventually European medicine for over 2,000 years. The primary compound eugenol, responsible for clove's characteristic aroma, remains in active clinical use in dentistry today. Zinc oxide. Eugenol cement is used in tooth restoration. Eugenol is applied directly to exposed nerve tissue during dental procedures for its anesthetic effect. And root canal sealers contain eugenol for its antimicrobial properties. Laboratory research has confirmed eugenol's antibacterial activity against over 20 bacterial species, including Staphylococcus aureus, E. coli, Salmonella, and Listeria monocytogenes. Antifungal activity against Candida albicans and other Candida species has been documented through membrane disruption mechanisms similar to those of carvacrol and terpenin 4-OL. Eugenol also inhibits biofilm formation in several bacterial species at concentrations below its minimum inhibitory concentration, meaning it disrupts the protective structure bacteria construct even when present at levels insufficient to kill the bacteria directly. The compound that kept cloves valuable along ancient spice trade routes at prices equivalent to precious metals is the same compound that dentists use in clinical formulations today. Number 18. Calendula. Calendula officinalis, the war flower carried by medieval soldiers for battlefield wound care, whose triterpenoid compounds have been confirmed in clinical trials to accelerate wound healing, and whose antimicrobial activity against Staphylococcus aureus and Streptococcus species makes it simultaneously a wound-healing and infection-preventing preparation. Calendula has been used for wound care and skin infections continuously across European medicine for centuries. It earned the name pot marigold in part from its documented use in preparations for skin conditions, and it earned the informal name war flower from its widespread use in battlefield wound management where access to formal medical treatment was unavailable. The triterpenoids, particularly faradiol esters, in calendula flowers have confirmed anti-inflammatory effects through inhibition of inflammatory enzymes. Allantoin contributes cell proliferation-stimulating activity that accelerates tissue repair. And the antimicrobial compounds in calendula, including multiple flavonoids and essential oil components, have documented activity against Staphylococcus aureus and Streptococcus species, the bacteria most commonly responsible for wound infections. Clinical trials have confirmed that calendula preparations significantly accelerate healing in burns and surgical wounds compared to standard care in several published comparisons. The cosmetic industry uses calendula extract in premium skincare products at considerable markup. The source plant blooms continuously from spring through frost, requires minimal care, and can be grown in any garden. The salve that medieval soldiers carried is available to anyone willing to grow the plant and spend an afternoon in preparation. Number 19. Plantain, Plantago major, the weed in every lawn and sidewalk crack whose aucubin and allantoin compounds have anti-inflammatory and antimicrobial activity, with a 2015 clinical trial finding that plantain ointment outperformed standard pharmaceutical treatment on second-degree burns, a result that would have generated significant commercial interest if it had emerged from a synthetic compound. Plantago major, the broad-leaved plantain weed found growing in every compacted soil environment on Earth, is one of the most consistently overlooked medicinal plants in existence precisely because it is so ubiquitous. Native Americans used it medically long before European settlers arrived. Traditional European herbalists prescribed it for wounds, insect stings, and infections. It was introduced to North American ecosystems through European colonization and spread so rapidly that Native Americans called it white man's footprint. Wherever Europeans settled, plantain appeared. The compounds aucubin and allantoin are activated when the leaf cells are broken. Crushing or chewing the leaf releases them into active form. Aucubin has confirmed anti-inflammatory activity through inhibition of inflammatory signaling. Allantoin promotes cell proliferation for tissue repair. Both have antimicrobial properties. A 2015 clinical trial published in an Iranian medical journal found that plantain ointment significantly outperformed standard pharmaceutical wound treatment on second-degree burns, a result that received almost no attention in Western medical media because the compound cannot be patented, and there is no commercial entity positioned to develop it. The first aid kit that follows you wherever you walk has always been there. Most people spend money having it removed from their lawns. Number 20. Yarrow, Achillea millefolium, named for Achilles, who according to classical tradition, used it to treat the wounds of his soldiers at Troy, containing achilletin and other compounds with confirmed platelet aggregation, promoting hemostatic activity alongside antimicrobial properties, simultaneously stopping bleeding and preventing the infection that follows, the most complete field wound treatment in the plant kingdom. The Latin name Achillea millefolium carries the entire history. Achilles, the greatest warrior of classical antiquity, used yarrow to treat the bleeding wounds of his soldiers at Troy. Whether that specific attribution is historical fact or mythological encoding of genuine medical practice, the documentation of yarrow as field wound medicine spans 2,000 years of continuous use across armies, healers, and indigenous peoples on multiple continents that had no contact with each other. The compound achilletin promotes platelet aggregation, accelerating the clotting cascade that stops bleeding. This hemostatic activity has been confirmed in controlled research. Yarrow measurably reduces bleeding time when applied to wounds. Simultaneously, antimicrobial compounds in the plant, including camphor, borneol, and various flavonoids, inhibit the bacterial growth that would otherwise colonize the wound once bleeding stops. The wound is closed and disinfected by the same application. Most antimicrobial plants perform one function. Yarrow performs both functions simultaneously in the same application. For anyone who spends time outdoors, knowing yarrow on site is practical emergency medicine with a 2,000-year field-tested record. The plant grows in every temperate meadow on Earth. Achilles knew it. His soldiers lived because of it. 20 plants, each one addressing infection through mechanisms that pharmaceutical antibiotics cannot fully replicate and that bacteria cannot easily evolve around. The antibiotic resistance crisis is real, it is growing, and the pharmaceutical pipeline for new antibiotics is inadequate to address it. These plants are not a replacement for antibiotics in serious infections that require clinical treatment. But they represent a category of antimicrobial knowledge that was functional for thousands of years before synthetic antibiotics existed, knowledge that was largely set aside when pharmaceutical antibiotics made it seem unnecessary, and that the resistance crisis is now making necessary again. These plants are still here. The knowledge of how to use them is recoverable, and the mechanisms they use to fight infection have not changed in the years since we stopped paying attention to them. Hit the like button if this changed how you see the plants around you. Drop a comment and tell me which plant surprised you most. Subscribe and ring the bell. I will see you in the next one.